Cristina Lazcano. She's an assistant professor in soil ecology and plant nutrition. Her lab, the Soil Biodiversity and Health Lab, is located at UC Davis in the Department of Land, Air, and Water Resources. From root microorganism interactions to ecosystem assessments, this lab is committed to finding ecologically based soil management strategies that will reduce the environmental footprint of agricultural production while satisfying the nutritional needs of the increasing world population. Her research includes relevancy from vineyards and tomatoes, cover crops, to grazing, no-till, compost, and more. So today we have the pleasure of having her with us, and I'm very much looking forward to getting to know more about your work and learn from you, Cristina. So thank you so much for taking the time to be here with us.
I was muted. That's kind of a classic of Zoom meetings. So thank you for inviting me. I really appreciate this opportunity to meet you and meet everybody, especially in these times where it's so difficult to really meet in person. So this is a great opportunity. I'm gonna share my screen and we can start. So in this presentation, I'm going to be talking about organic fertilizers, how they affect soil microorganisms, and what does it mean for soil health. And so before kind of jumping in, I'm going to talk a little bit about myself. I am originally from Spain, and that's where I was born and raised, and I did my undergrad and my PhD. And after that, I spent some time working in Denmark, and then I moved to California. It was already 10 years ago, so time flies. And I spent a couple of years at the University of Calgary in Canada doing a postdoc, and then I came back to California, and I've been here since then. So as you, Margaret, mentioned, I am a soil ecologist. So I study soil ecological interactions and how they affect ecosystem processes. For example, the cycling of nutrients or plant health and productivity, and how that also affects the quality of the environment. So most of my studies focus on greenhouse gas emissions and how they are affected by plant microbial interactions, for example. I study carbon sequestration in soils and the effect of ecological interactions also on water quality. And obviously, I'm really interested on how management affects this whole story, right? So a lot of my studies work are actually focused on management strategies like tillage, but mostly fertilizers. I have done a lot of work with organic fertilizers. So that was a bit of a background about myself. And since I joined UC Davis, I am actually very excited to know and see the interest that there is in soil health. And soil health has been, it's a concept that it has been adopted by, not just by researchers, but also by industry and growers and agencies alike. And this is because there is research that has been being published recently that shows that soils are important, not just for supporting crop production and crop quality, but also for regulating environmental quality and climate change and from climate change mitigation, right? So it's an exciting time to be a soil scientist and especially a soil ecologist. And if you have been attending grower meetings and research presentations, you've probably seen this definition of soil health already many times, but I'm going to go over it again, and especially because I want to make the connection with soil biota, which is going to come in the next slides. But soil health is defined as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So in summary, a healthy soil is a soil that functions and provides services. What are those services? There is a sort of different types of services that healthy soils provide that are related to more agronomic aspects. Like, for example, here in green, we have the support of high yields and crop quality. There's also services that are related to the soil ecosystem. So a healthy soil should have a high diversity of soil organisms, also microbial activity. There are services that are related to environmental quality, for example, they support the storage of carbon and also the regulation of greenhouse gas emissions. Some of other services are related to the cycling of nutrients. So a healthy soil should be able to supply nutrients, but also retain nutrients. And then there are services that are associated to water. So a healthy soil needs to have physical properties that allow for the movement of water, but also for the retention. And this is particularly important in arid areas like here in California, right? So there's a bunch of things that are expected from healthy soils, as you can see here. The concept of soil health builds on a more traditional concept of soil quality, but there are fundamental differences in what they are emphasizing. Soil quality might be more of a snapshot, a picture in time of properties of soil, for example, a certain amount of nutrients or a certain texture that are more static, right? And soil health emphasizes the capacity of soil to keep processes going that are actually providing for those nutrients, right? So there is an emphasis on the living component of soil, right? Soil health is really grounded and is really directly related to the fact that soils are alive, right? And they support living organisms. And if we talk about soil biota, then we have to talk about soil microorganisms because they have a direct role in many of these functions that we just mentioned. And this is another concept that you probably heard, the concept of soil microbiome, which it's a term that describes all microorganisms that are found in soil. And that includes not just bacteria, archaea, and fungi that are what most soil tests are telling you, but things like viruses, things like protists, for example, and other microbial eukaryotes like algae, right? So it's a bunch of things that are microorganisms and are living in the soil. When we want to study the soil microbiome, there's different things that we can look at. We can look at what we call the structure of the microbiome, and that's pretty much like answering the question of who is there, who's living in the soil. And then we can look at the function of the microbiome, which refers to what is it that those microbes are doing. And I want to kind of give you an overview of the different types of analysis that are out there and that are being used in ecological studies and the type of information that they provide. So for example, in the terms of soil structure, there's different tests that are commercially available. And all of these different tests, they provide different amounts of resolution, and they also have different costs. So here, from lower resolution to higher resolution and also lower cost to higher cost, we have microbial biomass carbon, which is a very kind of rough measurement of, it's just telling you the amount of carbon that it's contained in the microorganisms. And then with more resolution, we have PLFAs, which is something that you probably already heard at some point. And it refers to the analysis of the abundance of membrane lipids that are actually characteristic of different microbial groups. So this test provides a bit more resolution, but it's also a little bit more expensive. Now, for the highest amount of resolution and also for the highest price, you have tests that use DNA markers, like cysteine RNA, it's a genetic marker for bacteria and archaea, or ITS is a genetic marker for fungi. So those are giving you the highest resolution to study soil diversity. In terms of function, there's also different tests that we can look at with different resolution and different amount of information that they provide. The simplest test that we can use is the analysis of microbial activity that measures the CO2 that is released from microbial respiration. Then we have enzyme activity, which is telling us how active the different enzymes that are produced by microorganisms are in the soil. This is a little bit more expensive, but also provides more resolution. It's specifically telling you how those enzymes are working in the soil. And then all the way into the most costly, but also with the highest resolution, we can analyze the soil metagenomes, which is going to tell us the abundance of genes that are regulating microbial functioning, right? Okay, this is just for a little bit of background on the type of analysis that are out there. Going back to the idea of soil health, there are different principles that are usually followed to support and boost the health of soils. But by far, one of the most important is related to the increase of soil organic matter in soils, right? It's one of the main principles of management for increasing soil health. Organic fertilizers, for example, like manure or compost, are a direct input of organic matter to the soil. And so it has a direct effect on soil health. It affects the physical, biological, and chemical properties of soils that are normally associated to soil health, right? So organic matter, compost, organic fertilizers improve biological properties because they're a food source for the soil food web. They improve chemical properties because they increase cation exchange capacity and supply nutrients. They regulate and they sequester carbon and therefore regulate the climate in the long term. And organic fertilizers also improve soil physical properties by reducing bulk density and contributing to soil aggregation. So there's a whole array of benefits of organic fertilizers. And additionally, there's environmental associated to organic fertilizers, which is the fact that we are recycling organic waste materials. And so when those organic waste materials are recycled into organic fertilizers, then they are not disposed of in landfills. And so we can avoid the emissions of greenhouse gases from landfills and also the leaching of contaminants, right? Also, the use of organic fertilizers allows for the recycling of nutrients within agricultural systems, carbon, nitrogen, nitrogen, phosphorus, and potassium, as well as carbon. So there's a whole lot of benefits, direct benefits for soil health, and also indirect environmental benefits of using organic fertilizers. Now, the problem is that we're just talking about organic fertilizers, like this big category, but there's a tremendous amount of variability in organic fertilizer. There's a lot of different products that are out there available commercially, and all of them are extremely different physical and chemical and biological properties. Okay, so depending, for example, on the type of feedstock and processing degree, fertilizers can have differences in total carbon contents, but also dissolved organic carbon, nitrogen, phosphorus, and biological properties as well. And these are some examples of studies that are showing that fertilizers like compost, vermicompost, have bacterial and fungal communities than the original feedstock from where they're produced. So here, this study showed that the bacterial community of vermicompost and compost was completely different, but also different from the original waste that was used to produce this compost and the vermicompost. And something similar happens with the fungal abundance. Fungal abundance in this other study was very different from a compost and a vermicompost that was produced from the same cattle manure. So because of that, we can also expect that all of these different types of organic fertilizers are going to have different effects on soil health and crop yields. And so this is something I want to show you. It was part of my PhD. And this was an experiment that I designed to study the short-term effects of organic fertilizers compared to inorganic fertilizers on soil health by looking at the microbial community the yields and the quality of different sweet corn hybrids. Specifically, we wanted to look at how organic fertilizers affected the structure of the microbial community, the effects on soil nutrients and crop yields, and whether they were genotype-dependent responses to these different nutrient sources. So in this experiment, we used three different types of fertilizers. We had an inorganic source of nitrogen, phosphorus, and potassium, and we had two organic fertilizers. We had a rabbit manure. It's not very common, but at that point, that was what it was available regionally. So it's important to kind of use the organic sources that are regionally available, right? In this case, it was this huge rabbit farm that was producing a lot of manure. So we were looking at alternatives to recycle this. So that was the feedstock that was used to produce vermicompost. Those were the two organic fertilizers that were used. And the two organic fertilizers had very different contents of total carbon, but also dissolved organic carbon. Both forms of carbon, I mean, total carbon and dissolved organic carbon, were higher in the manure as compared to the vermicompost. And this is something that typically happens if you compare raw manure with a processed manure, which could be compost or vermicompost. These three types of fertilizers were combined to produce three types of fertilization treatments. So we had on one hand a 100% inorganic fertilization treatment, and then we had a vermicompost where 25% of the treatments of the nutrients supplied were supplied as vermicompost, a manure treatment where 25% of the nutrients were supplied as manure. And all of these were added at standard rates and also at a higher rate. And again, we studied the effects of plant genotype. And the reason for that is because there is evidence that plant genotypes interact differently with their environment because they produce different types of exudates through the roots. And so they interact with the microorganisms in different ways. So, and in addition to that, different plant genotypes also have different nutrient use efficiencies and resource allocation. So we included 4 types of corn hybrids, which were the result of the combination of 2 genetic backgrounds and 2 main mutations. So the genetic backgrounds provide most of the genetics for the hybrids, and the mutations were related to a single gene mutation that regulated the accumulation of sugars in the seeds. So sugary were it's what is known as sweet corn, and shranking is what is known as super sweet corn. So they had different amount of sugars in the seeds. All of these fertilizer treatments, the dose and the genotype, were combined into plots that were 10 square meters with 20 stands each. And the plots were arranged in the field following a randomized block design. We repeated this trial for 2 years, and at the end of the 2 years, we harvested the plants to measure growth yields and also nutrient contents. We took soil samples to study the microbial community. We studied the structure, so we used PLFAs for that, and we studied the function of the microbial community. So we used enzymes. We also assessed microbial respiration, bacterial and fungal growth. We measure the soil biochemical properties, nutrient contents, but also dissolved organic carbon and nitrogen. And we measure, again, in the different genotypes, we looked specifically at the microbial community in the rhizosphere, and we use PLFAs for that. So this is a bit of what we did. And now I'm gonna show you some of the results of this trial. So here in this figure, you see the rates of bacterial growth in the top and fungal growth in the bottom of the soils under inorganic fertilizer, vermicompost, or manure at the 2 different rates. So we had in gray the standard dose and in white the high dose. So as you can see, organic fertilizers promoted the growth of bacteria and fungi, and specifically the stronger effects were observed in bacteria. So organic fertilizers stimulated microbial growth in the short term. These were all experiments that lasted 3 months, so it was really short term. And in addition to that, when we looked at the specific group of bacteria that was stimulated by using PLFAs, we've seen that it was gram-negative. There was one type of bacteria that are known to feed on organic matter preferentially that was stimulated. That was gram-negative bacteria. So we thought that this was probably related to the input of organic carbon. When we looked at the function of the microbial community, we did that by looking at the enzymes, activity of the different enzymes. So we studied protease activity, and this is an enzyme that is related to the cycling of nitrogen. Protease breaks down proteins that contain nitrogen. We studied the activity of beta-glucosidase, which it's related to the cycling of carbon. Beta-glucosidase breaks down glucose. And then phosphatase, which is directly related to the cycling of phosphorus, breaks down phosphorus compounds. So we've seen that in all of the 3 enzymes, addition of organic fertilizers increase this enzyme activity significantly. So what it's telling us is that vermicompost and manure increase nitrogen, phosphorus, and carbon cycling as compared to the inorganic fertilizer. So both aspects of the soil microbial community, the structure and the function, were affected. We looked at the yields of the crop. And the most interesting thing is that we saw a lot of variability across all of the different hybrids. And so I'm going to explain this graph a little bit. We have here results for plant biomass and yields. And for the 4 types of hybrids that we studied, we have the sugary hybrids. I'm going to use the laser pointer. So we have the 2 sugary hybrids here and the 2 shrunken hybrids. So some hybrids, especially sugary hybrids, responded in a positive way to the addition of organic fertilizers. And sometimes those effects were more related to the type of organic fertilizer. So manure had a stronger effect on plant biomass in these 2 genotypes, for example. Whereas some other genotypes seem to respond negatively to the addition of organic fertilizers. They seem to reduce their yields. We looked also at the quality of the grain. And here in this figure, again, you have the 4 different hybrids, the 2 sugary and the 2 shrunken. And this is showing different rows are showing different things. So the first row shows the content of nitrogen in the grain. The second one is showing the grain yield, which is the amount of yield of grain per corn cob. And the last row is showing soluble solids, so bricks. And again, we saw a lot of variability. Some hybrids show positive responses to the organic fertilizers, increasing nitrogen contents in the grain, increasing grain yield. And some others actually saw decreases in grain yields and also in soluble solids. So the next question is, why is this happening? Why are different hybrids responding differently? And so we went to look at the rhizosphere. And here this is a discriminant analysis of the 31 PLFAs that were identified in the rhizosphere samples. The different so here, like U determines sugary hybrids, H shrunken, I it's for inorganic, B vermicompost, and M is manure. Solid lines represent standard doses, and dashed line high doses. So the first thing that we noticed is that there was a clear difference in the microbial community between the 2 main types of hybrids. Sugary and shrunken hybrids had different microbial communities. Within each type of hybrid, the microbial communities were differentiated by the type of organic fertilizer and the rate. So the different hybrids were responded differently to the treatments. Okay.
We have a quick question in the chat.
Yes.
Someone is asking, what are the axes?
The axis in the chart. This is not like, this is a multivariate analysis. So what you need to look at here, it's the proximity of the different data points within the graph, which they're gonna tell you how similar the different data points are, right? So these circles are actually, these are soil samples represented, and they are showing similarities in the microbial community composition. So the closer together, like over here, these ones are overlapping. That's showing that the microbial community in those soil samples was very similar. If they are further apart, that's showing that the microbial community was more different. But this chart is summarizing the diversity of the microbial community using 31 PLFAs. Not sure if that was clear enough.
Thank you.
All right. Thank you for the question. All right. So kind of like wrapping up this section, we saw that use of organic fertilizers supported microbial activity and promoting the growth of microbes, also the cycling of nutrients and plant nutrient uptake. The effects were variable and depended very much on the type of organic fertilizer and the degree of processing. There were stronger effects observed with manure, which were possibly directly because of the higher DOC, which is dissolved organic carbon. And we also saw plant genotype dependent effects on nutrient quality and nutrient uptake and grain quality, which were associated to differences in the rhizosphere microbial community. So going back to the properties of healthy soils, in this trial, we observed that there is a direct effect of organic fertilizers on the biological component of soils. Organic fertilizers also support yields and crop quality, and also they are directly responsible for the cycling of nutrients, right? So they actually kind of affect a lot of this and drive a lot of these properties of healthy soils. But again, the effects are very much dependent on the chemical composition of the organic fertilizer. The last thing I want to talk about in the minutes that we have left, it's how organic fertilizers actually affect the environmental outcomes of these practices. So I'm going to focus on the production of greenhouse gases from organic fertilizers and how they can be balanced with the storage of carbon. This picture here is showing you what happens when we put organic fertilizers in soils and how the carbon it's stabilized in different ways. And I'm going to explain it a little bit. It shows a little bit, there's a lot of information, things to unpack here. So once organic matter, organic fertilizers are incorporated into the soil, all of it is unprotected initially. And there's different things that are going to happen here. So part of that organic matter, it's going to be too difficult to break down. That's going to be organic matter with low litter quality. And because of that, it's going to remain in the soil without being broken down by the microorganisms. So what's going to happen is that organic matter is it's going to be what we call biochemically protected. The rest of organic matter, it's going to be processed by the soil microbial community in some point, right? After that, then microorganisms will produce residues, products of the composition, which are going to interact with the mineral components of soils. So part of those residues are going to be incorporated into aggregates. Part of them are going to be absorbed into silt and clay. And so that's what we call physical stabilization of organic matter. So all of these things can be happening in the soil when we put organic matter. if you notice, at the same time that carbon is being stabilized, there's always a slight release of CO2. So every time we put organic matter in the soil and organic matter is going to be broken down, there's going to be a release of CO2 just because we are stimulating microbial activity. So that on one hand. On the other hand, when we put organic fertilizers in soils, organic fertilizers can have different amounts of nitrogen. The nitrogen, it's going to be processed again by microorganisms. And in this chart, I'm showing you the processes of nitrification and denitrification. So organic matter, when it's mineralized by the soil microbial community, is transformed into ammonium. Ammonium is transformed into nitrate through the process of nitrification. Both ammonium and nitrate are plant available forms of nitrogen, right? So this is a very important process. And then nitrate can be subjected to denitrification, all right, which is a transformation of nitrate all the way into N2. Now, these two processes are also sources of another greenhouse gas, which is nitrous oxide. So these are natural processes that are happening in soils, but that can be a source of a greenhouse gas. So the majority of the gases that are produced from soils are mostly CO2, but there can be a certain proportion of nitrous oxide that is being released from soils. And even though the proportion of nitrous oxide is much, much lower than the one of CO2, the nitrous oxide is particularly important because the global warming potential, it's actually much larger than the one of CO2. So in the end, it's going to be a really important driver of greenhouse gas emissions and global warming, right? The global warming potential of nitrous oxide is almost 300 times larger than the one of CO2. And we know already that soils and agricultural soil management is the largest source of nitrous oxide, right? So there is a cost which might be associated with applying organic fertilizers. And we've seen that previously. This chart is from a study that we did in a dairy manure farm where they were growing corn in the Central Valley, and we've seen that there was a clear association between fertilization events with liquid manure and releases of nitrous oxide. So in this case, the application of liquid manure led to large emissions of nitrous oxide right after. So it was very clear. So how can we avoid this? Generally, we need to follow the same principles as we follow for inorganic fertilizers. So adopting the 4R strategy, using the right source of fertilizers at the right rate, at the right time, and the right place, right? So using fertilizers carefully. For example, if you think that there's going to be, if you are afraid of losing nitrogen or emitting nitrous oxide, then you might not use a liquid manure. Maybe you decide to use compost, which has less amount of available nitrogen. Now, the challenges to this is that, as I mentioned, there's a large variability in the chemical composition between different organic amendments. So manure has a different chemical composition than compost, than vermicompost, right? So you need to understand that. Even within the same type of organic fertilizer, the variability can be really large between different farms. And this is something that is showing, this figure here is showing changes in concentration of ammonium, of different forms of nitrogen in liquid manure between 3 different farms in the Central Valley. So the concentration, for example, of ammonium can vary quite a lot, right? Another challenge is that mineralization rates seem to be very different between, and they seem to change a lot. So in the short term, in less than a year, the mineralization from compost, after it's applied to the soil can change from 5% to 22%, right? It's a lot of variability in mineralization rates. So what can you do to overcome these challenges? The first thing and the best thing you can do is know your fertilizer. Know the chemical composition of your fertilizer. Once you know that, specifically focus on nitrogen, and what you need to do is reduce the amount of plant available nitrogen that you are putting into the soil in each fertilization event. And plant available nitrogen, it's the sum of ammonium nitrate and the organic nitrogen that it's going to be mineralized over that period of time. Things that you can do too, combine fertilizers which have different amounts of plant available nitrogen and also mineralization rates. So you can match the release of nitrogen to the uptake of your crop. Another thing you can do is promote the association of plants with beneficial microorganisms. And this chart is showing it's from a study that we did a few years ago, studying the effects of mycorrhizae on nitrous oxide emissions in a compost amended soil. So we've seen that mycorrhizal fungi, when they were associated in tomato plants, were able to reduce nitrous oxide emissions from the compost amended soil. So promoting this type of interactions might help reduce nitrous oxide emissions in soils that are organically managed. And then obviously manage your soil in a way that you can promote these beneficial interactions. So reduce the intensity in tillage, for example. You can use microbial inoculants. You can apply mycorrhizal inoculants to your crop. And then consider synergies and interactions with other healthy soil management practices. So we have seen in other studies that combining compost application, for example, with cover crops, can reduce nitrous oxide emissions. So stacking and overlapping practices can help reduce the losses of nitrogen. And I'm going to wrap up, just going overall to kind of remind what we discussed in this presentation. So we've seen that organic fertilizers can improve soil health and support crop yield and nutritional quality, but the effects are very dependent on the chemical composition of the organic fertilizers and specifically the content of organic nitrogen and carbon. Large amounts of carbon, available carbon and nitrogen can trigger nitrous oxide emissions and reduce the environmental benefits of organic fertilizers. So we need to manage organic fertilizers carefully, following the 4R principles to reduce losses. And another thing which I think is really interesting, and I started to explore a little bit further in my research, it's this interaction between plants and microbes, and which are actually telling us that there might be certain genotypes or varieties of the same crop that are better for organic production. And so we need to study a little bit more about what are those traits that allow plants to profit more and to benefit from soils that have highest amounts of organic matter or more microbial diversity, for example. All right, and so with that, thank you for your attention, and I can take any questions.
Thank you so much,